All right, ladies and gentlemen, welcome back. So we are going to continue working on these cladogram practice problems, but this time, rather than me just giving you data, we're going to figure it out for ourselves, and then use that data to construct the cladogram. So we're still working on molecular differences, and in this case, we're looking at a hemoglobin protein sequence, so amino acids, and we're going to compare human to the rest of these species. All right, so first thing I want you to do is to go ahead and take a look and compare across each amino acid. Where do you see a difference that isn't human? All right, and then we're going to highlight those differences. Okay, so if I take a look across that very first amino acid, all right, and I want to highlight those differences, so let's turn my highlighter on here. All right, if I come across the top, chimp is the same, gorilla has the same, but that monkey is different. So I'm going to highlight that one. All right, and then the horse also has the same. I go down through that second amino acid. They're all identical across all species. Same with the third. Same with the fourth. All right, these are all the same. Same here. So here's a difference. So we're going to highlight those both in the gorilla and the monkey, but the horse has the same one. Here's another difference. Here we've got a difference. There's another. This one's also different. Okay, so if I go back through, just do a double check. Okay, and I think we found them all. Okay, so now I'm just going to count up how many differences I see, and we're going to record those over here. So if I go human to human, well, there shouldn't be any differences. They're the same species. All right, and so the number of differences that I have there would be 0. I take a look down the chimpanzee column, and I didn't highlight anything there either. All right, so there actually aren't any differences, at least for this particular protein, for this portion of that protein, so they're also going to get a 0. I go down that gorilla and I see 1 highlight, so that means there's 1 difference. Monkey, I've got 2, and then the horse, I've got 4, it looks like. All right, so there's the data that we're working with. So now I can use this to actually construct my cladogram. And at this point, I've really simplified the table here for you just to kind of get the information that we needed based on that data that we just collected. So now I'm going to go ahead and figure out where these guys are going to fit on the table from each other. And again, we're going to use the biggest number first. Those are the ones that have to be the furthest apart. And so in this case, it's the horse and the human. So just knowing a little background, I'm going to put the human up here. All right. And that means the horse comes down here. Okay, so I've taken care of human and horse. So now I can work in either direction. I can either go smallest number from the human or the biggest number. And let's go ahead and just do the biggest. So the next biggest number would be that 2 for that monkey. So that means that guy's going to come here. All right, so I've taken care of that. Gorilla's got 1, so he'll be next. And then the chimp also has 0, so he'll be right here. All right, so let's take a look at this data and just answer a couple of questions. So according to the data and according to the cladogram that we built, which species is most closely related to the human, and how do you know? Well, if we take a look at the table, right, the organism that would be most closely related to that human would be the one that has the fewest number of differences between the sequences, and thus would be the closest on the table, or closest on the cladogram. So who's right next to the human? The chimpanzees. All right, let's take a look at the next one. Which species is least closely related to the humans, and how do you know? All right, again, if I'm going to look at who's least like the human, that means they have to have the most number of differences between their amino acid sequences, and as a result, they would be furthest from each other on the cladogram. So in this case, that's the horse. All right, so let's go back to the actual sequence differences that we looked at here. All right, and this one's just asking what is the difference between the human and the gorilla hemoglobin? So if we look, the only difference here was that one that we highlighted. In humans, they have an RUG, which is arginine, and in a gorilla, they have a lysine, which is a LYS. What might be responsible for that change? Well, remember that the flow of information in your cells goes from DNA to RNA to amino acids, right? So the original set of instructions for everything that your cells do is housed in your DNA, but that DNA is housed in your nucleus and never leaves. So we make that RNA copy of just the piece of information that I need. That RNA message goes from the nucleus out to the ribosomes, which are your protein factories in your cytoplasm, so outside of the nucleus, inside the cell, and that's where we build the amino acids. So the DNA is the set of instructions to know which amino acid to put in which order. So if I change the amino acid sequence, like that lysine from that arginine, that means something had to change in the rest of these sequences. So if I change this amino acid sequence, the RNA sequence changed, which ultimately means the DNA sequence changed. So what do we call a change in DNA? It's a mutation. So somewhere along the way, as those gorillas and those humans were evolving and they're branching off from each other, diverging from each other, some sort of mutation occurred in the genetic structure of what would ultimately become humans to make that arginine, where in the gorillas it was the lysine. All right, guys, so we are going to try the same thing, but now we just have a little bit more data. So we're now looking at a cytochrome C amino acid sequence, so a different protein, and I'm looking at the differences between a handful of more species. Humans here in the middle, though, that's going to be what we're comparing everything to. So we're going to do the same thing we did last time. We're going to come through and we're going to highlight the differences that we see. So what's not like the human, okay? So let's work through this. All right. So there are our number of differences. So let's go ahead and count up how many we see. So if I take a look at the horse, we've got, let's see, 1, 2, 3, 4, 5, and 6. And so I'm just going to write that at the top here. You guys have a table to write this in. The chicken, 1, 2, 3, 4, 5, 6, and 7. Tuna, 1, 2, 3, 4, 5, 6, 7, 8. And this one actually is one as well, the blank there, so 9. Frogs, 1, 2, 3, 4, 5, 6, 7, and 8. Sharks, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, and 13. Turtles, 4, 5, 6, 7, and 8. Monkeys, I've got 2, and the rabbits, 1, 2, 3, and 4. Okay, so now that I've got the data, now I can start building that cladogram, and it's the same idea for everything else that we've done. So take the opportunity to try it yourself. Okay, so if we take a look, right, I'm going to put the human up here because that's ultimately who we're comparing this against. All right, so I'm going to take care of that human. And then the biggest number of differences is between that shark. So that means that shark has to be the first one down here. All right. The next biggest number is my tuna. So that tuna is going to be the one that comes next. Okay. Next number is 8, but check it out. I've got a frog and a turtle with both having 8 differences. So what does that tell you? Remember, that means they've got to be branching off of the same branch. So they branch off of each other, but come off the main line together. So that's going to include my frog and my turtle. All right. Next biggest number is the chicken with 7. All right. Then comes the horse at 6. The rabbit with 4. And then finally that monkey with only 2 differences. And there you go. Okay, so let's take a look at some follow-up questions then. So according to your analysis, according to the cladogram we built, which species is most closely related to humans, and how do you know? All right, so again, if we're saying most closely related, that means they have to have the fewest number of differences between their sequences, which also would mean that they're closest to each other on that cladogram. So in this case, it should be your monkey. And then which species is least closely related? And how do you know? All right, so if we're going to say least closely related, that means they have the biggest number of differences between their sequences, and so they should be furthest apart from each other on the tree. So in this case, that would be your shark. All right, again, let's go back to the sequence itself and which 2 species have the closest sequence to each other. And again, it's going to be that human, that monkey. If I take a look across those sequences, they only had 2 differences, hence why they're most closely related to each other. And so that's where that sequence match comes from.